realize that we are all learning about molecular targeting together. As we learn, we have to also understand that the science is constantly evolving. As scientific discoveries are made, they can directly change or impact practice, not just in the discovery of new drugs, but in how we better use the drugs that are already FDA approved. When learning about a new drug, I find it helpful to ask 3 questions. What is the target? What is the role of the target, both in cancer development and in the normal function of the body? And finally, how does this drug hit the target? We will try to answer these questions as we proceed through this program. Scientists are discovering new ways to target oncogenesis, and many of those discoveries are being translated to the clinical setting. In this particular program, we are going to focus on the specific targets for which we currently have FDA-approved agents. We will be discussing the following molecular targets: tumor cell receptors, such as EGFR, HER2, PDGF, c-KIT, and BCR-ABL. We will discuss the process of angiogenesis and endothelial cell ligands and receptors like VEGF and VEGFR. We will discuss signal transduction pathways such as mTOR, tumor-specific antigens such as CD20, CD52, and CD33, apoptosis and enzyme complexes such as 26S proteasome, and finally, multi-targeting. Tumor cell receptors, such as receptor tyrosine kinases, are known to be dysregulated in a number of cancers and serve as intriguing targets. One of the receptor families we know the most about is the family of human epidermal growth factors, the HER family. There are 4 members of this group of receptors: HER1, HER2, HER3, and HER4. HER1 is also known as the epidermal growth factor receptor, EGFR, or it may be referred to as CERB1. It has 3 names, but they all refer to the same molecule. I liken it as to going by my given name, Teresa, but also answering to nicknames like TK or TJ. HER2 was the first member of the HER family to be specifically targeted for cancer therapy. Epidermal growth factor receptors are normal proteins that regulate cell growth, differentiation, and survival. Growth factors bind with specific growth factor receptors located in the cell membrane and initiate the transmission of signals to the cell nucleus. This process, as we have already discussed, is called signal transduction. Most growth factor receptors act through the tyrosine kinase pathway, or TKP, for signal transduction. Human epidermal growth factors and receptors are found in many cancers, including breast, colorectal, head and neck, pancreatic, and non-small cell lung cancer. When overproduced or dysregulated, they can be associated with poor prognosis and serve as a prognostic marker for risk of tumor relapse and decreased survival. When looking at the HER family, please note that all of the receptors have tyrosine kinase activity except HER3. Also note that HER2 does not bind to growth factors. HER2 is activated by overexpression. Let's focus first on the epidermal growth factor receptor, EGFR, also known as HER1 or CERB1. What role does EGFR play in cell signal transduction? First of all, a ligand binds to the receptor site. Epidermal growth factor, or EGF, as shown here, is one of the ligands of EGFR, but there are also others. The receptors dimerize, which, as you remember, means pairing of 2 single receptors or monomers. The intracellular tyrosine kinase region is activated, which causes binding of the ATP molecule region, generating phosphorus for receptor phosphorylation. Phosphorylation signals intracellular pathways which stimulate the cell cycle. Of note are pathways such as RAS and RAF and MEK, which can actually be targets in and of themselves. Drugs in clinical trials are designed to hone in on some of these intracellular targets. Expression of EGFR and its dysregulation is common in lung, colorectal, and head and neck carcinomas. It is seen in virtually all squamous cell carcinomas. EGFR dysregulation correlates with increased metastasis decreased survival, and poor prognosis. Its activation of the tyrosine kinase phosphorylation signaling process leads to tumor growth and development through enhanced proliferation, invasion, angiogenesis, metastasis, and the inhibition of apoptosis. EGFR protects tumor cells from cytotoxic effects of chemotherapy and radiotherapy, making these treatments less effective. EGFR is expressed in normal healthy cells from all three germ layers, particularly those of epithelial origin, such as skin, liver, and GI tract. It also produces cell factors, such as vascular endothelial growth factor, for example, that promote angiogenesis and further encourage cellular proliferation. As shown in this slide, a ligand binds to EGFR, and the receptor dimerizes, moves into a matched pair. Once dimerization occurs, the activation of the tyrosine kinase domain proceeds, sending signals to the nucleus of the cell that regulate cell growth. EGFR may homodimerize, which means it matches to another EGFR, or heterodimerize, which means it matches to another HER family member. Let's look at a second member of the HER family, the member called HER2 or C or B2, which also has a tyrosine kinase domain, but you'll note it has no ligand. It is activated by overexpression and plays a large role in breast cancer. HER2 is a proto-oncogene that is amplified not only in breast cancer but also in a number of human cancer types. It is a cell surface receptor involved in growth regulation by sending signals to the nucleus through the tyrosine kinase intracellular signaling pathway. When HER2 protein is overexpressed on the cell surface, it is the result of gene amplification, meaning the cells have extra copies of the HER2 gene. HER2 overexpression is not hereditary, and 20 to 25% of breast cancers are HER2 positive. While we currently have molecularly targeted agents toward EGFR or HER1 and HER2, the other two members of the family, HER3 and HER4, are under investigation. Two other important tyrosine kinases are c-KIT and BCR-ABL. When a protein called c-KIT develops a gene mutation, it can become too active and send out a signal to tell tumor cells to keep growing. Tumors, such as gastrointestinal stromal tumors called GIST, are characterized by cell surface expression of the dysregulated c-KIT tyrosine kinase protein, which is also known as CD117. When c-KIT is mutated and dysregulated, it does not depend on a ligand to activate the intracellular tyrosine kinase signaling. BCR-ABL is a protein tyrosine kinase formed when there is a gene translocation, also known as the Philadelphia chromosome cytogenetic abnormality. This translocation is seen in 95% of patients with chronic myeloid leukemia, CML, and 15 to 30% of adult patients with acute lymphoblastic leukemia, ALL. The BCR-ABL oncogene is formed when the BCR gene on chromosome 22 is fused with the ABL gene located on chromosome 9. The BCR-ABL tyrosine kinases can cause dysregulation of intracellular signaling, resulting in enhanced proliferation and resistance to apoptosis of myeloid cells. Here is a schematic showing the BCR-ABL kinase and its intracellular signaling pathway. While most of our molecular targets involve tumor cells, there are other ways to target the growth and spread of cancer. One important target is that of angiogenesis, growth of new blood vessels, and the role of the endothelial cell in the process of angiogenesis. Angiogenesis is a normal and necessary process in our bodies. It is needed for the formation of new blood vessels and for many physiologic functions, such as embryonic development and wound healing. As our bodies need new vessels, they can form from pre-existing capillaries. However, in the process of malignant tumor growth and metastasis, angiogenesis can become pathologic. There is a loss of balance between positive and negative control over the growth of blood vessels to the tumor. Many human tumors express vascular endothelial growth factor, a ligand known as VEGF. We know that tumor survival and growth depend on an adequate blood supply for oxygen and nutrients. In fact, tumors cannot grow beyond 1 to 2 millimeters without a blood supply. VEGF is the primary angiogenic factor produced by tumor cells. In fact, one of the results of EGFR expression or dysregulation in tumor cells is the production of VEGF. VEGF can also be expressed by tumor-associated stromal cells such as fibroblasts. Tumor blood vessels are abnormal and depend on a constant supply of VEGF for maintenance. While VEGF plays a major role in binding to VEGF receptors on endothelial cells, it has also been discovered that VEGF receptors exist on tumor cells, protecting tumor cells from hypoxia, chemotherapy, and radiotherapy. Disruption of VEGF can prevent tumor growth and may even cause the tumor to regress. There is a family of tyrosine kinase receptors called vascular endothelial growth factor receptors, VEGFR1, 2, and 3. And their ligands, VEGF-A, B, C, and D, that play an important role in the process of angiogenesis and tumor cell survival. The signaling pathways that lead to angiogenesis begin when VEGF binds to VEGF receptors. This causes the intracellular tyrosine kinase domain of the receptor to be phosphorylated. A signal transduction cascade follows, which causes several local events to occur. The blood vessel develops fenestrations, or holes, that allow proteins and serum to leak out into the surrounding tissue. VEGF induces production of an enzyme called matrix metalloproteinase, or MMP, by endothelial cells. The MMP leaks out through the blood vessel holes or fenestrations and breaks down interstitial collagen. These two events cause the creation of a gel-like matrix just outside the capillary that is an ideal substrate for new blood vessel growth. VEGF also stimulates the proliferation of endothelial cells, which can then follow the matrix and form a new blood vessel. Let's turn our attention now to intracellular pathways and processes. There are endless possibilities for targeting intracellular signal transduction pathways. One signaling pathway of particular interest in renal cell cancer and other solid tumors is the mTOR pathway. It can be called the center of multiple signaling. As you can note in this slide, the mTOR pathways may be stimulated by a variety of signals, from familiar receptors like estrogens, growth factors, and cytokine receptors to integrins. Integrins are adhesion receptors that play a role in cell movement, adherence, and survival. mTOR stands for mammalian target of rapamycin, and it is a serine-threonine kinase. It is downstream of the PI3K pathway and is a major signaling hub. mTOR controls nutrition in the cell and serves as an intermediary in a variety of cell signaling events, such as cell growth, proliferation, angiogenesis, and cell survival. It is easy to see why mTOR is a target of much interest in treating cancer. A potential target for cancer therapy lies in particular antigens that exist on cancer cells. In the arena of hematologic malignancies, this type of targeting has become a mainstay of cancer treatment. Three surface antigens in particular play important roles in the treatment of lymphomas and leukemia. The antigen CD20 is expressed by normal and malignant pre-B lymphocytes and by mature B lymphocytes, but not on normal plasma cells. CD20 is not found on hematopoietic stem cells or in other normal tissue. Greater than 90% of the B-cell non-Hodgkin lymphomas express the CD20 antigen. The antigen CD52 is present on the surface of B and T lymphocytes, a majority of monocytes, macrophages, natural killer cells, and a subpopulation of granulocytes. A proportion of bone marrow cells, including some CD34 positive cells, express various levels of CD52. The antigen CD33 is expressed on the surface of leukemic blasts in more than 80% of patients with acute myeloid leukemia, or AML. CD33 is also expressed on normal and leukemic myeloid colony-forming cells, including leukemic monogenic precursors, but it is not expressed on pluripotent hematopoietic stem cells or on non-hematopoietic cells. Another important target, particularly in the arena of hematologic malignancies, such as multiple myeloma, is that of apoptosis through honing in on enzyme complexes within the cell. A proteasome is an enzyme complex that is found in all cells. The 26S proteasome functions to break down cellular proteins in both healthy and cancerous cells. They can be thought of as an intracellular recycling center. The 26S proteasome breaks down ubiquitinated proteins, which are proteins that have been specifically tagged for degradation. In cancer cells, the ubiquitination process is in overdrive, so an increased amount of proteins are tagged for degradation. The ubiquitin proteasome pathway plays an essential role in regulating the intracellular concentration of specific proteins, thereby maintaining homeostasis within the cell. Cancer cells depend on protein regulated by the proteasome for proliferation, metastasis, and survival. While single molecular targeting has proven useful in the treatment of cancer, certainly we realize that cancer cells can learn to circumvent the inhibition of only one target. Thus, agents are being developed that can hit more than one molecular target at a time, increasing the possibility of an agent's effectiveness. This particular slide depicts the variety of targets that are available for molecular targeting. As we get into presenting information about specific targets, we will show how even a single agent may have the ability to hit multiple targets. So now that we know about the variety of molecular targets that can be exploited, let's turn our attention to how drugs are developed to hit those targets. The treatment of cancer with molecularly targeted therapy represents a shift in how we approach cancer treatment. As opposed to chemotherapy, which non-selectively attacks dividing cells in order to destroy the cancer cell, targeted therapy involves drugs that target specific tumor molecules or processes. As we have discussed thus far, targeted therapies can be designed to hone in on very specific molecules or pathways to help moderate, control, or kill cancer cells. Drugs designed for targeted therapy include two main types of agents: monoclonal antibodies that hit targets in the extracellular domain, and small molecule agents that hit targets within the intracellular domain. Monoclonal antibodies tend to be large molecules that cannot cross into the intracellular domain. All of the FDA-approved monoclonal antibodies currently used in cancer treatment are given intravenously. In our discussion, antigens are normal cell surface proteins or ligands that serve as targets for binding engineered monoclonal antibodies for the treatment of cancer. Ideal target antigens for cancer treatment should have certain characteristics. They should be specific to tumor cells or processes like angiogenesis that play a role in tumor growth and can be found minimally on normal cells. They should be located on the surface of the tumor cell so the antibody can find and get to the target antigen and not shed into the bloodstream. They should occur in high numbers, and they should play a role in tumor cell survival. Significant differences exist between chemotherapy and monoclonal antibody therapy. Monoclonal antibody therapy is specifically aimed at tumor cells expressing a target antigen. A more favorable side effect profile is seen with some monoclonal antibodies compared with chemotherapy. The side effect profile of monoclonal antibodies is related to the activation of the immune system, the location of the target antigen, and the type of monoclonal antibody. Side effects from monoclonal antibody therapy are not cumulative. Some monoclonal antibodies sensitize tumor cells to chemotherapy and overcome chemotherapy resistance, and the anti-tumor effect of monoclonal antibodies may be delayed and prolonged. Conversely, chemotherapy is directed at killing all cells dividing at certain cell cycle phases and is non-selective. Chemotherapy adverse events are systemic, involving all rapidly dividing cells and the body's response to toxic drugs. Finally, side effects from chemotherapy may be cumulative and can lead to long-term complications. The function of an antibody or immunoglobulin is to recognize an antigen and then interact with other serum proteins to eliminate the antigen or the cell associated with that antigen. The Y shape of the antibody has two active sites, FAB and FC, and two activities. The FAB portion of the antibody is the antigen binding site that recognizes and binds to a specific antigen. The FC, or constant region, is the end of the antibody that signals the immune system to destroy the cell the antibody has bound. The binding of antibody to antigen is highly selective, rather like a lock and key fit. When an antibody selectively binds to a specific antigen, the antibody-antigen complex acts as a flag or target, drawing other immune cells to the bound cell to destroy it. A monoclonal antibody recognizes only a single unique antigen and is produced by cloning a single cell. Hybridoma technology, developed in 1975, allowed for the mass production of monoclonal antibodies. Monoclonal antibody production is illustrated in this slide. A mouse is inoculated with purified protein, and antibody-producing cells are isolated from the mouse spleen. Antibody-producing cells are mixed with myeloma cells, which are immortal cells unable to produce antibodies. Finally, only antibody-producing cells that have fused with myeloma cells are selected. The monoclonal antibody is then engineered and produced in large quantities. After the monoclonal antibody specifically binds to the target antigen on the cancer cell, it induces inactivation or destruction of the cancer cell through one or more possible mechanisms. It can cause neutralization of tumor cell growth by direct interference with normal biologic activities of the antigen, such as signal transduction of cell growth messages. This cytostatic process can slow growth of the tumor cells. An example of this is when a monoclonal binds to a receptor and moves it into the cell, a process called internalization. A monoclonal antibody can cause promotion of antibody-dependent cell-mediated cytotoxicity, ADCC. The FC portion of the bound monoclonal antibody recruits effector cells, such as phagocytes, T cells, and natural killer cells of the immune system, to release cytokines that destroy the target cell. Complement-dependent cytotoxicity, CDC, which activates the complement system, a cascade of naturally circulating blood proteins, is a way for a monoclonal antibody to also destroy cells. Finally, direct induction of apoptosis, or programmed cell death, can cause the antibody-bound cell to self-destruct. Monoclonal antibodies are designed in the laboratory to bind to a single target, such as specific molecules on cancer cells. In fully human antibodies, the entire antibody has been engineered to contain only human antibody gene sequences. Human antibodies are usually named with the suffix UMAB, UMAB. Murine monoclonal antibodies are derived from mice. Mouse antibodies have a very short half-life in the human body. They are not as effective as human antibodies in eliciting an effector response from the CDC and ADCC system. And they can cause the development of human anti-mouse antibodies called HAMA that neutralize and render the mouse antibodies ineffective against the tumor. Mouse antibodies are usually named with the suffix MOMAB, M-O-M-A-B. Chimeric monoclonal antibodies end with the suffix XIMAB and contain both human and mouse gene sequences. Typically 70% human and 30% mouse. I think of the X as a clue that these drugs are a cross between human and mouse. Humanized monoclonal antibodies ending in ZUMAB also contain human and gene mouse sequences. However, they contain more human sequences than do chimeric monoclonals. Typically, humanized monoclonals are 90 to 95% human and 5 to 10% mouse. When monoclonal antibodies are named, not only is the suffix a clue to their makeup, but one can tell what they target by their name. If a generic monoclonal has a TU in the name, the target is on the tumor cell. Monoclonal antibodies target antigens or receptors on the cell surface that extend into the extracellular domain. They can also target ligands such as growth factors that circulate in the extracellular domain. Monoclonal antibodies can be naked or unconjugated with nothing attached to them. This slide is an example of an unconjugated monoclonal antibody attaching to an antigen or receptor on the cell surface. Like naked or unconjugated monoclonal antibodies, conjugated antibodies target specific antigens. However, they also introduce piggybacked anti-cancer agents directly to tumor cells, such as biologic or chemotherapy agents, toxins, or radioisotopes. Although agents attached to conjugated antibodies are targeted specifically against tumor cells, nearby healthy cells can still be negatively affected, especially if radioisotopes are used. If the specific target antigen is found on normal cells, the conjugated antibodies can cause damage to these cells as well. While monoclonal antibodies tend to be large molecules that attach to antigens in the extracellular domain, small molecule agents can cross the cell membrane and hit targets that abide in the intracellular domain. Examples of small molecule agents include kinase inhibitors, proteasome inhibitors, and immunomodulatory agents. Unlike monoclonal antibodies whose generic names end in MAB, you will note that these agents end in different suffixes, like NIB, MIB, LIMUS, and MIDE. One of the largest subtypes of small molecule agents is the category of kinase inhibitors, which can include tyrosine kinase inhibitors and serine threonine kinase inhibitors. Tyrosine kinase inhibitors, or TKIs, prevent phosphorylation of tyrosine kinase. As you will remember, phosphorylation is the attachment of a phosphate to a protein and is a very important step in cellular signal transduction. Tyrosine kinase inhibitors are small molecules that can cross the cell membrane of a tumor cell to work in the intracellular domain. They directly interfere with the catalytic activity of the tyrosine kinase by blocking the binding of ATP. These drugs generally can be given orally, and the generic names of these drugs end with the suffix NIB. Here's another example of how a tyrosine kinase inhibitor can block the signaling cascade. In this particular example, the cell is an endothelial cell instead of a tumor cell. In this example, you can see how some tyrosine kinase inhibitors can actually block tyrosine kinase at multiple locations within the cell. These agents are called multi-kinase inhibitors. Tyrosine kinases can have both advantages and disadvantages. One of the advantages is that they actually can work on receptors that may be mutated and do not contain an extracellular domain. This is something that could not be accomplished with a monoclonal antibody. They also can cross the blood-brain barrier, and unlike monoclonal antibodies, they have a very low potential for immunogenicity. These drugs do not require visits to the clinic as often as monoclonal antibodies since they are given orally. However, on the flip side of that, there may be issues related to drug compliance because they are dosed orally. Finally, because most tyrosine kinase inhibitors are metabolized by the hepatic cytochrome P450 pathway, the potential does exist for drug-drug and drug-food interaction. Earlier, we discussed that the serine threonine kinase mTOR is an important target in many cancers, particularly because it is the very center of so many cellular signaling pathways. Thus, small molecule agents that target mTOR in the intracellular domain are being used in renal cell cancer and are being investigated in many other solid tumors. You will remember that a proteasome is an enzyme complex that is found in all cells. Proteasome inhibitors are novel agents designed to block the function of proteasomes. Proteasomes function to break down the majority of cellular proteins in both healthy and cancerous cells. We likened this function to a recycling center for proteins. Proteasome inhibitors block the proteasome from functioning, causing a buildup of proteins which affect multiple cellular signaling cascades. When the signaling pathways are disrupted in cancer cells, it can lead to cell death and inhibit tumor growth. Finally, there are small molecules called immunomodulatory agents that play a role in the treatment of cancer. While the exact mechanism of action is not known for these types of agents, one example being thalidomide, it is felt that they work in a variety of ways through immunomodulation and inhibition of angiogenesis. We live in an ever-changing world where scientific discoveries about molecular targets guide our treatment decisions for patients with cancer on a daily basis. Whether aiming at targets through monoclonal antibodies or small molecule agents, molecularly targeted therapy has incredible and unlimited potential and has become a mainstay of cancer treatment in both solid tumors and hematologic malignancies.
Thank you. Please stand by.